The question that I want to focus on this time is talking primarily about cancer and a little bit about stem cells. Right? So, first thing that you need to be thinking about when you see stem cells is what actually is a stem cell. So a stem cell is an undifferentiated cell. So that is a type of cell that can go and turn into any other type of cell. Most questions that have something to do with stem cells will have that sort of thing in it. Right, following on from that, it says figure 4 shows a malignant tumour in the trachea of a patient. Right, remember the trachea is the tube that's going down from the mouth down towards the lungs. Give one way in which the malignant tumour differs from the benign tumour. Right, remember a benign tumour is a tumour that's not really going to cause any problems, that isn't going to cause any damage, that isn't going to spread. A malignant tumour will cause significant damage and does have the potential to spread. So the next part, it says scientists can treat a patient's tumour by replacing the trachea with a plastic trachea. The plastic trachea has a layer of the patient's own stem cells covering it. Right? Then it goes through how the stem cells from a patient can be transferred to the plastic one and such forth. So then, it says in step 3, the cells are left for 48 hours to divide. Name this type of cell division in step 3. Right, it's just normal cell division, so it's mitosis. Remember, the other type of cell division that we get is meiosis, but meiosis only occurs when making sex cells. In step 3, the cells are given oxygen and water. Name 2 other substances that the cells need to divide. Right, just think about what cells actually need to grow. Right, so think about what might be included in that liquid growth medium. So glucose. Think about what's needed for cell replication. So when a cell's replicating, it needs producing new proteins. So you might need protein in there. You might need amino acids in there. So in a question like that, you need to be thinking about what does a cell actually need to grow? What does it need to divide? 4.5. Give 2 advantages of using the stem cells from the patient and not a dead human donor. First one, stem cells will go from a live donor, will go and they'll proliferate more, right? So they'll coat it a lot quicker. Stem cells from the person themselves won't be rejected by the immune system, so you are less likely to get an immune response against them as well. You might, in some cases, talk about the fact that some people don't think that it's ethical. However, I would only go and mention anything about ethical reasons if you really can't think of anything else. 4.6. So sometimes the stem cell trachea is not strong enough. Doctors can put a stent inside the trachea. It's just how the stent in the trachea helps keep the patient alive. Right, first thing you need to do, think about what a stent is. Right, when you go and put a stent down, it's a wire mesh that opens up and holds it open. That'll be the first part. The second part comes from this suggesting how it helps keep the patient alive. Well, it helps keep the patient alive because it holds the trachea open. Therefore, gases can keep moving through it. Oxygen can get into the patient, which they need to live. Right, the final part of this question talks about stem cells can also be obtained from human embryos. Evaluate the use of stem cells from a patient's own bone marrow instead of stem cells from an embryo. Give a conclusion for your answer. Right, on a question like this, it's really important that you talk about the advantages and disadvantages for each. What are the advantages of human embryo stem cells? What are the disadvantages? Same with stem cells from a person. Right, after you've gone and written a few advantages and a few disadvantages for each, put some sort of conclusion. It doesn't matter which one you come down on. as long as it's there. All the examiner wants to see is that you are thinking, that you are articulating what you think. Right, go back, any parts that you're still unsure about, watch that part of the video again, get your specimen paper and go and have a go at this question and try and really concentrate on that squeezing every last mark out of it.